Instagram too. Hello,
Hello.
welcome.
Welcome to Philosophising. We thought we'd try
Where
something
we
different today, where Nathan and I did not share what we prepared with each other, like at all.
because we're gonna fight.
Yeah, it'll be a gentle debate. It'll be like a chatting debate.
Yeah, yeah, yeah. I mean, I
um,
feel like the format is pretty similar to how we talk about stuff like in the stream normally, but um,
yes, that's true. It is not that
mhm.
different from how we normally would chat and talk about science with each other anyway. But well, basically what happened was Nathan sent a Google link and didn't share it with me, and so I didn't have access to it, and so I prepared on my own, and he prepared on his own, and then we're like, wait, today we're debating population coding and labeled lines, which are I guess is cool. And then I was just like, yeah, I feel confident about doing this without seeing what you're gonna say, and then so
I don't
we
feel like
thought it
I
would
actually
be
said that, but we're good. Hang on, let me also just check the audio is working. But yeah, we so what we are going to be talking about today is how the brain works, and in particular some like different theories about like the fundamental way that the brain is organized, and some of the debates that have been happening about that.
no.
So I
And
put it. Oh, go
I would
ahead.
like to preface it with that I don't think there is a absolutely correct answer. I think there's examples of both. But like I think it's also really fun to think about what people have
Hmm.
done like to experimentally like prove and disprove either side of this debate. And that's really cool, right? Like that's a like it's progress in science when we are reached a point where we are debating and like finding the nuanced, like it's like this most of the time except for this, or it's like this in most instances except for this, and like those exceptions are really cool. Like it helps us figure out how the brain works.
Yeah, and I think I think it's such a really interesting question too, because it really gets at this question of kind of like, okay, what are the fundamental underlying principles of like how the brain works? Like you can see, like we call this stream, how does the brain actually work? And because I think that's really what it's getting at is like with like all kinds of computers we have some sense about like, okay, here's its architecture. And this is really a question about like, what is the fundamental architecture of how our brains and nervous system works?
Yeah, and what are like the limits of it, right?
Hmm.
I feel like some of the arguments that I will make today is quite limiting because of the literal resources our brain has. Um,
Oh, yeah. Hmm. Yeah, I
well,
was going to ask what you meant by that, but I guess we'll get to it.
oh yeah, okay, so
Yeah,
we can pick
yeah.
sides now. I
Or actually,
think
do we want to maybe start with like just like an introduction to kind of what this is, like what we're talking about? Like, what
oh
are
yeah.
the labeled lines versus ensemble coding?
Okay. Yeah. So like I guess like the most like simple version of what labeled lines is kind of um like when sensory information, well in the sensory field it makes as an easy example,
Hmm.
it's like every neuron in this labeled line receives the input from one type of sensory information, and then it just in sequence goes like first order neurons, the second order neuron, and a third order neuron, and at each level they tag another neuron and they bring that information into the brain, and then it gets processed as that sensory modality. So, for example, if you get touched, like in the foot, and that foot neuron, that sensory neuron that innervates your foot, will activate. Will activate a sensory neuron in your spinal cord. Will activate a neuron in your brainstem, and then in your thalamus, and then into your cortex, and then it'll basically tell into the cortex part of your brain where it represents your foot, and you will perceive that as your foot was touched. Like it's like it just
And
follows the same like uh yeah, it's like it's like a really simple, straightforward labeled line. Like
yeah,
every neuron in that order is does the same thing. It's like
I
that's I
guess
guess it's
kind of
not the
like,
most extreme version. Yeah.
yeah. And I guess kind of like as a more cognitive person, kind of the way, and tell me if like this is not actually what you're talking about, because I think it's important to like define it. But like, I've always kind of thought about this. as it's kind of saying like that we have neurons that act, for example, like the light on your computer that tells you when it's charging, or the check engine light in your car, or something else. So it's like the function of those lights is to kind of represent and communicate one kind of information, and kind of fundamentally what this, so like when you have your check engine light in the car, you can say fundamentally the purpose of this circuit is that it tells you that it detects a problem with the engine. And that is how we know whether there's a problem with the engine or not. I guess kind of that's how I've always thought about it. So it's kind of in the brain, this is like how we know if we're touching something or not, or how we know if we're happy or not. Like there's these kind of dedicated like cells that, and I feel like in the most extreme version, it's kind of like, okay, if
It
you're
does
really
one thing.
happy,
Yeah.
yeah,
It does
it's
one
like
thing.
these
So
cells fire a lot when you're really happy, and they don't fire when you're sad, and then that is representing your current level of happiness.
yeah, and I guess the example in sensory would be like these neurons only fire when you eat something bitter, and then you're like
Mhm.
yuck, sad. So like literally all the way up from the primary sensory neuron that expresses the bitter receptor on the periphery, like you get a something nasty touches that ending on that neuron, all the way into the brain, into the like the bitter cortex area. It just every neuron along the way says bitter shit, bitter shit, bitter sucks, bitter sucks, bitter sucks, yuck. And at the end it goes yucky taste. And it all the way from periphery all the way in, it does the same thing. And it like doesn't change its tune. And it's, that's the label line.
Right.
It
Yeah, that makes sense. And then, so the alternative to that that people talk about, I feel like that is more confusing. Like it's this idea of ensemble coding, right? And I guess kind of like, I guess kind of like the classical example of that would probably be something like a more complex smell or taste or something, where it's like, and I think this is something where it's not really debated, but it's like we don't really have any cells that specifically detect the taste of asparagus. Like we have kind of a whole bunch of different cells that all react to different compounds that are in the asparagus, or like, and even like different, with the same molecule, different cells will like bind to different parts of it. And so kind of it's actually this pattern over kind of a couple hundred different kinds of neurons that represents what does this taste like.
Yeah.
Um,
Like the combination matters more than the specific like one thing. So like in the case
mhm.
I said bitter, but like usually we don't just eat a pure bitter compound. Usually you're drinking something like coffee, which has sweet things in it and bitter things. And then like the combination of all these different sensory things will come in and combine to create this percept that you just consumed coffee. And it's not just bitter bad, like one thing.
Right.
It's like, oh, here's this like complex experience that includes like some sweetness and nuttiness and like different kinds of olfactory components, and all like creates this like little orchestra thing that gets perceived into a like a hoo-ha that is coffee perception.
Uh-huh. Yay, coffee. Yeah. But like I feel like it can be like I feel like it can be even for one object too. Like it's like even if you're just like, hmm, that smells like wine. Like it's like it can be like kind of you think about it as a single object, but it can be represented pretty in a pretty complicated way, I think is the other part of it. I
Yeah.
think like also like these are examples in sensory processing, but I think this also extends into like more higher cognition too, these questions. So like another part of this debate has been kind of, for example, like when you have a memory of a person, do you have like a group of neurons that represents their face, and kind of that like you have a neuron that's like dedicated to one person and it fires when you recognize that person, or does that person occupy kind of a multi-dimensional space over that's there's a whole bunch of features that are encoded by different neurons, and kind of it's this pattern of firing that represents them.
Yeah.
And
And there's evidence for both, right? Like I think
yeah.
famously there's like a Jennifer Aniston area in the brain, like, or
Uh-huh.
like like Jennifer Aniston neurons. And you're like, wait, why does it do that? And I was just like, well, every time we show them a picture of Jennifer Aniston, like these neurons fire, or like the face of some other person, like these neurons fire.
Mhm.
That's, in my mind, that's kind of an example of labeled lines.
Yeah. Although I will say, like, so we when I took a cognitive neuroscience class, we talked about the problems like with this, and I think one of the challenges to the labeled lines theory is that often we don't really know what neurons are representing. And so my professor actually told me this story where they had monkeys and they had electrodes in the left temporal lobe, which is where a lot of these neurons that seem to respond to specific people and specific objects are. And so they'd show them a picture of a face. They'd be like, some neurons would just fire only when they saw a face, and then they'd say, okay, so there's face selective neurons, and then they'd show them like a monkey, and they'd be like, okay, there's monkey selective neurons. They're different neurons. They seem like they don't overlap at all. And then they showed them like a picture of a mop, and then they
It was a
found, and
toilet
then
brush. It was a toilet brush.
yeah, okay, they showed. Oh, so you also
Specifically
heard
a
this
toilet.
story, yeah.
Of course,
Okay,
I
yeah. So
know
they
who did
showed
this
them this
experiment.
this brush, yeah. And then they also saw neurons that were responding to that. And that was kind of what made them start to rethink this because they're like, okay, it has human selective neurons. That makes sense. It sees humans a lot. It has monkey selective neurons. Makes sense evolutionarily. And then they're like, wait, it has brush selective neurons. And then actually, like, then when people kind of started doing the math, what they realized is that if the neurons in these areas were really organized by like this, like you couldn't have enough neurons in your brain to recognize all the objects that we see. Um, and
That
so
was
to make a long
kind of what I was
story
alluding
short,
to.
yeah.
Oh.
what they
Like there's not
what
enough
they
resources for all the things
oh
in the
okay,
world that
yeah,
you need to
yeah, yeah.
represent. Yeah.
Uh-huh. Yeah. And then but I think what's interesting is kind of if you do a certain kind of experiment, these neurons look like they're encoding one thing. Like they look like they're encoding a face. But you never know, like, okay, are they also encoding something else? And is it kind of, I think the people who study this often call this like a latent space, where it's like maybe these neurons encode stuff you tend to see in the morning. And that's like a specialization that they've encountered with experience. And like, it seems like we actually have these kind of latent spaces in the brain, but it's really hard. You can't test every single possible stimulus. So I think oftentimes you'll see neurons that you think are specific for like 10 years until someone tests some other kind of stimuli and they're like, oh, actually, it also responds to that.
Yeah. Okay. So my explanation I got from reading that exact same paper, is
Mhm.
that the toilet brush, it's specifically a stick with, like, the thing that you clean the toilet bowls
Yeah.
with.
Mhm.
So it has a circular center and it has hairy edges, and so
Mhm.
the labeled line explanation for this would be it activated the circle with edges, which is important for this animal to know. And so faces of other monkeys and humans, as well as this toilet brush, all activate this labeled line that is general, like circular thing with hairs. And
So the
so
theory is kind of that the labeled lines are just like not tuned that specifically.
yeah, so like usually when we look for like super like label lines, like we give them stimulus like orientation, like bars, like light bars of light and like different angles, orientation of light,
Hmm.
like different brightnesses of the light, and then you can like get really nice like orientation tuning, and you can like figure all that out. But when it comes to things like faces, there's not enough neurons in the world, so you can like do you know what I mean? Like if you know a lot of
Hmm.
people, there's not enough neurons to represent each person separately. And
Hmm.
so
Right.
I know this is also going against my labeled line theory because that means there's no actual Jennifer Aniston. It's just like blonde lady, blonde
Hmm.
lady face. But it's still like is fundamental to like how I think the brain works, which is like we kind of funnel them to like kind of sort of it specific things, and then the rest is like sort of like hand wavy. Like the funneling it into like almost mostly hairy round things with like hairs coming out of it being a face and a being that
Hmm.
matters. I mean, sometimes it's just good enough. And that's that's how our brain works. It's just barely good enough to do the thing, and it's not perfect.
I mean,
And
that
that's
makes sense to me, but like, but we can tell the difference between Jennifer Aniston and a brush, right? Like people perform above task on above chance on differentiating those things.
That's true. I mean, if you use only that one neuron, if you're only recording from that one neuron and trying to use it to build a model to be like, is it Jennifer Aniston or not? Neuron would do very poorly.
Yeah. I guess like, and I guess like the thing I am getting at is kind of, I think this is one of the reasons why I kind of lean towards the idea that these things are distributed, at least in most cases, is there's this question of like what does the like how do you get an interpretation of the stimulus out of it? And so if you have individual neurons that are representing a thing, that kind of makes sense. But it's like if you have these kind of fuzzy representations, then something has to be looking at all these different neurons and saying like, okay, because it's space shaped and it is a familiar actress, it's Jennifer Aniston. And it's like, so it seems like that interpretation system is really using a kind of labeled line encoding.
Yeah, and then we're
I'm
into the
saying not a labeled
calculation.
line
No,
encoding,
it's not
an
using labeled
ensemble
line anymore.
encoding.
It's using
Yeah,
ensemble
yeah,
encoding. Um,
yeah.
I the real answer is it probably is somewhere in between where like all the information
Mhm.
coming in comes in labeled lines so that it makes some sort of sense, and then you have to do ensemble coding to make to make sense of it all the way to create the percept. Okay, then
Mhm.
do you think animals that we are like their level of consciousness depends on how much they diverge from just labeled lines? Like
Hmm.
do you think
Mhm.
animals that are like, let's start with like not vertebrates to barely vertebrates to like full on like vertebrates mammals, like do you think like a sea like a I was gonna say a sea anemone, but actually yeah, sure, a sea
Yeah,
anemone.
like something that's really just like reflexes.
Yeah,
Like,
it's like super simple in reflexes and poke at withdrawal, like those kinds of like things. Like you can argue like those are very labeled liney, right? Like like it doesn't like
mhm,
it's like it senses bad chemicals, it goes away. It senses bad thing, aversion runs away. As opposed to like a human where we're like, oh, bad things, and you're like, let's evaluate if this is actually bad for us. Like maybe we'll run towards danger instead.
let me think about it. Yeah,
Yeah,
yeah.
like like we
Uh-huh.
can make that evaluation. So we're like
So
on
like
the other
you're asking
spectrum.
like, is there a difference between like animals that can be masochists and animals that cannot be masochists, and whether they use labeled lines?
Sure, yeah, like or how much of
Yeah.
their like maybe
Hmm.
it's like every creature is like a split. Like I maybe humans are
Hmm.
like a 60-40 split where we're like 40% labeled lines and then the rest of it is population coding, or maybe we're like and then like maybe the sea anemone is like 90-10 split where
Hmm.
they're 90% just labeled lines and then like 10% is being like, oh, I like the fish I ate today. Do you know what I mean? Like instead of like survival, like the line between like we must have this so that we survive and reproduce and make the future generations of my creatures versus the like conscious like experiencing of the world, like and the
Right.
difference is actually how much ensemble coding exists and how much um just straight labeled line boring analog exists in this
Yeah, I
animal.
mean, I think that's true in like I mean, like we talked about consciousness a couple streams ago, and like I mean, there's all these ideas about like complexity of recurrent processing and like abstraction and things like that. And I guess like my thought is like, okay, if something is mostly just like labeled lines, it probably doesn't have much of the infrastructure needed to have consciousness, because it's like we talked about, right, like how consciousness seems to involve a lot of feedback processes, and it doesn't, like if you have this kind of feed forward labeled line system, it's not clear why you would have those feedback processes, or like, would you need them for anything? Like maybe for
Yeah.
attention still, I guess. Like, but
Yeah.
um,
And then is that even peak performance? Because we talked about this really early on, but isn't peak evolution like crabs? And like crabs have some of the most labeled liney circuits in like crab, you know what I mean? In crab neuroscience, like you can literally patch this neuron in every single crab, and like you do this thing, and then it twiddles their like left foot, their left, you know, do you know what I mean? Like it does the exact same thing in every single crab, and it's so stereotypical, and it's like mapped out. There's a name for every single neuron in their entire thing, and neuroscientists have been doing this, right? Like we create connectomes of the entire C. elegans, and the fly, and now we're working on creating the entire connectome of a mouse, and we're like, wait, the entire connection or making of this experiment or making of these connectomes rides on the fact that labeled lines exists, right? Because if it's just completely different from mouse 1 to mouse 2 or crab A to crab B, like we just have a map of crab A. Do you know what I mean? Like we don't have a map of
yeah,
all crabs and how all crabs do the thing if every crab does it differently. Oh, sorry, right?
I
Like,
mean, I don't know. I mean, I imagine like that you could have. I kind of feel like it would still work because you could have convergence at some level, but it could also be not necessarily a labeled line, right? Like it's you can have a part of the brain that encodes escape behavior, and maybe the way that it encodes escape behavior is different in every single individual, and even like different cell types participate in different individuals. But because it outputs to the motor system and makes you jump, it is encoding escape actions kind of regardless of what it's doing internally.
got it. So you think like the input in, like the first part's input in, and then the motor output out, those parts are like labeled and like stays exact same, but like the black box is everything else that happens in
I
the
guess
brain
like
could be different.
I guess like what I'm thinking is like it kind of makes sense to talk about. I guess what I was thinking is mostly it makes sense to talk about the connectomics, even if there's a bunch of differences between individuals. Like I don't think, and I kind of wonder if like those are the exact same thing. So it's like one is like, okay, is there labeled lines, which like I kind of think about that as like very sparse encoding. It's like this neuron encodes something specifically. And another one is, are those consistent between different individuals? And I think those are not necessarily the same question. Like, I
Oh,
think I could have, I mean, it's kind of like the inverted spectrum argument, right? Like, what if my red is actually a different
then
perception
you're
from
red.
your
Yeah.
red? Like, and it's like, that could be true regardless of whether it's a labeled line or whether it's an ensemble coding thing.
Yeah. I think the only good experiment, like I'm trying to think of good experiments that can
Like, how would
like
you experiment? Like,
yeah, yeah.
just,
So
yeah.
so I know there's like perceptual tasks that you could do. So like if okay, so there's like a color task that humans can do where we can be like, is your yellow my yellow? So basically if you take I think like a like cyan and magenta, or like the two colors, I forget. Like the
Hmm.
combo of the two colors, you get yellow. Is that right?
Hmm.
Like the light
Yeah,
spectrum.
that
So
sounds right.
yeah, and then you take you could basically imagine you have two like spotlights, and each of them have a dial on them, and then you can like basically dial how much magenta and how much cyan color, and you have two little dials, and then they focus into a center, and it creates a yellow spotlight. And so then you give someone a like a color card and be like, here, this is yellow sunshine, whatever color, like a like a paint color. And then you tell them, and you take away the paint color, and you're like, okay, you remember that color, go make that color. And so they can
Hmm.
dial the cyan and the magenta until they can get to the yellow that they perceive like was the yellow that just they just saw. And I think my yellow and your yellow is going to be different 100%. And I think it falls on like a normal Gaussian, how much
Hmm.
of each
Like it's kind
is
of
given.
a,
Yeah, yeah. But it's not like completely off kilter. So this is the question of like, is your red my red, right? Like, is your green my
Right?
green? Like, if we, so this is like, is your yellow my yellow? Because that yellow is a perception. It's not an actual yellow. It's the perception of these two wavelengths like mixed together to create. Do you know what I mean? The percept.
Right.
And,
And this
and
experiment suggests that it is, but like it's kind of an approximation, I guess, is what you're saying, right? Like
correct. Like
it's
it's close. It's not a completely different wavelength. It's just we're off
Mhm.
by that much.
Uh-huh.
And like we're off by that much, which means it's good enough because in real life, who cares if your yellow is off by that much? It's like yellow is yellow.
Right. Yeah. Yeah. Hmm.
um
That's interesting. Oh yeah, FFG293 says I didn't know you could actually prove people are seeing the same color. Yeah, I didn't
yeah,
either.
like this
Yeah.
is like the only experiment I can think of where like people have really tried to like
Hmm.
tackle it, and I've it's really fascinating, right? Like these are really interesting ways to think about if you're perceiving the same thing as I'm perceiving. And this is like this
Hmm.
happens all the time, right? Like differences what of my perception of things different from Nate is a really interesting like thing because like we could go out and have a meal and eat the same dessert and I'll be like that dessert was way too sweet and Nathan will be like
Uh-huh.
that was tasted amazing, it was perfect, right on
Uh-huh.
the money. And now like this could happen all the time, right? Like and it's just like what is like what we
And it's
perceive.
like, yeah, and it's the question about like, okay, are these differences about like did it actually taste different to me, or do I just kind of have different like reward associations with things that taste like do I value sweetness differently than you do?
Yeah,
Like I feel like that's the really hard question is like, is my subjective perception of it different, or just do I have different experiences with things that taste like this?
yeah, and it's hard like they
Yeah.
do this is an issue that happens in like wine tasting a lot. Like
Hmm.
it's really rare for someone to calibrate them, and I think that's why it's important for the people that do these kinds of things to like recalibrate or go to similar schools and like calibrate and be like, here we're when we taste this we're going to call this blank amounts of astringency, or like this means it has this much on the nose, or do you know what I mean? Like they have to
Hmm.
recalibrate
Yeah. Yeah.
themselves because they have to all call it the same thing so that they can communicate about it.
Yeah, and they do that, right? Like
Yeah.
they learn like kind of all the different adjectives that you use in new practice. Like someone will be like, "Oh, this line is like a 3 on astringency," and then you kind of learn how to be
Yeah.
consistent with that.
Like in some things it's more simple, like you could just like mix 50 grams of sugar and then like 60 grams of sugar, 70
Hmm.
grams of sugar, and then like go down the row and be like, like just practice until you can be like that's 30 grams of sugar, that's 60 grams of sugar.
Yeah.
And then like like really map it. We could do that, but at the end of the day, like the perception we're off by that much, right? Like we'll know when I say there's more sugar and there's less sugar. I think the scale is in the same direction. I don't know if it's inverted. It's ever inverted. Like when I consume 60 grams of sugar, I'm gonna be like, oh, that's sweeter. I'm pretty sure 60 versus 30. I'm sure Nathan's gonna be 60 and 30. 60 is sweeter than 30. But like if we had to taste 100% grams and like none,
Yeah.
like we would block
I
it
mean,
differently in between that scale. Do you know what I mean? Like,
yeah, and I think like that's kind of the really the philosophical argument that people talk about, which is like so they say like, okay, if I genetically modified you so that your sweetness receptors are using like a different GPCR, so it's actually inhibited when there's sugar and excited when there's no sugar, you would learn to describe things the same way as I was, but would it actually be the same experience for you?
yeah. So we have
Yeah.
done these experiments. We actually have done them. So
Oh, really?
funny that you pick sweetness as this. So
Hmm.
all along the sweet pathway. So you have like the taste
Hmm.
receptors for like glucose. And then like, second order neurons and then TS, like you can literally follow every order of neuron all the way up. So if you
Hmm.
just like knocked out these sweet receptors and replaced it with bitter,
Uh-huh.
this animal will drink bitter water. It will drink bitter water as much as it would drink sweet water like other animals would drink sweet water. And it's because it just like activates that part of their brain.
Yeah.
Isn't that wild?
Which
And
makes sense because it has no error signal, right? Like there isn't any
yeah,
feedback that is saying like, hey, actually that wasn't sweet. Like you
yeah.
made a mistake.
Yeah, and in fact, if you they don't even have to drink anything. You could literally just like electrically or optogenetically or using dreads activate the sweet portion of the brain, and they will lick the air. Like they will just be like, yeah, sugar. And, like, they will just, do you know what I mean? Like that's
I
how
guess
labeled
that makes
line.
sense. They're like, wow, this air is very sweet in here.
um like that's why it's so convincing. But to be fair, these are really rare examples where it's like really, really labeled lines. Like, and that's why I think it's, it's very like innate and like old, like an ancient system. It's like
Yeah.
survival. Eating sweet things makes you stay alive. Like, let's keep this in our brains. Like, it's like a conserved circuit because it's so important for survival. Like, even if you're like half brain dead, you should be able to do this. Like, do this every time. Keep doing it to
Mhm.
survive. um and there's probably redundant things and like ensemble coding and whatever to make it like
Yeah.
reinforcing, right? Like, what, like, remember how to get to the sweet water, like do all the other things so that you can get to the, get that hit of sugar again.
I guess
uh
I guess for me, like I believe that, but I also kind of I have a little bit of a problem with like the way that like kind of the philosophical part in it, and that kind of I think like it's very easy to say, okay, these neurons are like encoding sweetness, but that's different from like having an experience of sweetness. Like I guess, and the way that I would put this is like, okay, so if you took all the parts that we kind of know are labeled lines, these systems that record to like the sweetness, and then you disconnected them from the rest of the brain, and then you put sugar on them, like would you say the animal is having an experience of sweetness? And like, I wouldn't. Like I think like the weird thing to me about this when we're talking about these labeled lines is that to actually be generating an experience, something has to be actually labeling the lines. And like something like it needs to get like there's more processing that has to happen for it to be like a subjective sensation. And it seems to me like that is a process that involves kind of like fundamentally that's a process that has to do with like what is this neuron connected to and kind of how are those systems interacting is what
oh,
defines whether we really perceive it as sweetness or not.
okay, so you're saying like the circuit existing itself is not enough to be like that's a labeled line,
Like
because
I think
it's
it's a
connected
labeled
in that way.
line. I think it's a labeled line in some sense. Like if your definition of a labeled line is like we can decode whether you are eating sugar by recording this one neuron, it's a labeled line. But like I don't think it's a good explanation of how we perceive sweetness. Like if we just say sweetness is this neuron firing, and like that doesn't really make sense as a mechanism.
Got it. Like,
But I guess the way I was really thinking about this actually, so that's the sensory version, but the version that I of this that like actually bothers me is like when you talk to a lot of like popular science people do that where they'll be like serotonin is a neurotransmitter that makes you happy. And
yeah.
like how does it make you happy? I mean, it doesn't. There's this assumption they're making that serotonin is somehow has a label for happiness in it, and I'm just like, that's not really what's going on. Like, all serotonin does is it changes how neurons respond, and the reason it changes your mood is because it's doing these really complex alterations in how all these different brain networks are working.
I love that you bring this up because this is actually what I want to study right now.
Okay, yeah,
Yeah. So I moved to the periphery where things kind of sort of made
yeah.
sense into the brain recently, and
Uh-huh.
I was just like, wait, shit, things don't make sense anymore because of all the things we've mentioned. But when it comes to monoamine and neuromodulators, um, the labeled line version of this story for me is who expresses the receptor, because you can dump all the dopamine into the brain. If there's no dopamine receptor, it can do shit. Same with the
Mhm.
serotonin. Like, so in my case, it's which of the neurons express these receptors and in which context. So like in this context, these population of neurons express a lot of this serotonin receptor, and so when there is a release of serotonin, these neurons are the ones that are like labeled and they're activated, and then they're part of the happiness circle. And so it's not just every neuron everywhere, it's just the neurons that express the receptors for it. And so those are the neurons of interest, and that's why
But like, there's a lot of neurons that have serotonin receptors that have nothing to do with happiness, right?
Yes, absolutely. You're like, no, and so that's why I spent all these hours staring at atlases with like single cell sequencing and like looking at where all these receptors are, and I'm just like staring. It's like reading smoke. You're like, why are there a whole bunch of these receptors like specifically in this part of the brain and over here and not here, and like why is it here? Like, do you know what I mean? Like that is the nuanced part of figuring out the circuits that are involved. Is you got to figure out who's it's like the currency. Like serotonin is like the currency, and like if you're in America, you gotta use dollars. Do you know what I mean? And like if the
Yeah.
neurons are like Canadian, they're like, we don't take American dollars, we take Canadian dollars. Like get here some other way. And so they have to like tag like 4 other neurons using serotonin and then some other transmitters, and then eventually get to the Canadian dollar, and you have to like pass it along the way. And that pass along the way, that circuit, it looks like it could look like ensemble encoding because we don't understand it yet. There's too many neurons. We don't understand. And so right now it's like, there's a pattern where like the dollar goes into francs, or wait, francs is not a thing. It goes into a euro goes into like like it makes like 4 different transfers, and eventually then there's an increase of dopamine in this little population of neurons specifically here. And you're like, wait, how did they get there? And you're like, well, it had to do like 4 transfers. four tags before it got there. And meanwhile, all of these neurons are firing. And so if you're just looking at neurons and you don't care about what they're releasing and who they're tagged to, then it can look like ensemble recording. It'll look like a population encoded situation. But in reality, there's actually like circuits that were like really quick microtransactions that are happening. Be like, hey, like my Canadian dollar, like for three American dollars, swap that with this many pesos, and then this gets swapped with, do you know what I mean? Like that, all that shit's happening along
Right.
the way.
So you were saying it kind of looks like when you look at these systems, like there's a whole bunch of brain regions or cells that just kind of represent happiness as this really complex distributed pattern, but it would make more sense kind of if you understood like what receptors are on each neuron and like how they're connected to each other.
Yeah, like you
And
can
then
follow each like connection theoretically and make some sense of it eventually. Or there's like no hope for us neuroscientists. Like we would never solve any problems. We would never figure it out if they're just like all I mean, people are solving it in different ways, but I feel like there is some hope in this direction that I'm going for.
I guess, and maybe this is because I kind of come from a different field of neuroscience, but I guess like the part that I'm kind of skeptical about is like that the neurotransmitters are the important part. Like, I think like, and to me that seems kind of central in that it's like saying like when people talk about this like happiness or motivation as a labeled line system, like the idea is like, okay, like for example, like dopamine is a labeled line for like motivation to go engage in like to activate a behavior, for example. And like I guess I kind of struggle to believe that's the important part. Like I think it's more about what does the dopamine actually do that's the important part. Like what's really like it's like motivation, I would say, is really about like you're disinhibiting certain parts of the motor control system so that they can drive the muscles. And that can be done with dopamine, and that's how it's normally done, but it can also be done with like electrical stimulation or other kinds of drugs. And like to me, like the important part is like this pattern of activity. And sometimes like even the balance of activity between different parts of the brain. Like there's this idea that actually being outgoing is related, like kind of like kind of behavioral activation, going out and doing something requires having more output from your left prefrontal cortex than your right prefrontal cortex. And there's this idea that there's these competing behavioral inhibition and excitation systems. And like, to me, it seems like the real deal is like these systems are competing and mutually inhibiting each other, and they use some chemicals to do that. But the chemicals aren't really the important part. It's this balance of activity between these regions.
Yeah, as opposed to like specific neurons in the circuit that are involved in this
Yeah.
activity. Yeah,
Mhm.
I mean that's like this is the great debate, right? This is like
Yeah.
and that's why it's on philosophy. Like there are people on both sides, and on it's like a spectrum of like where we believe it could happen. I think it's you have to design different experiments to test each one of these, like to
Mhm.
test if this circuit matters for blank. You need to find, do you know what I mean, like you find the different variables to test that. Whereas if you just go in with different techniques to find, like just like record from a large group of neurons, you could end up recording, like I want to say like oscillations, which
Mhm.
like I feel like this happens, right? Like people that study
Yeah.
oscillations are like, this is it. Like this is what like this is the kind of activity that is the result of, you know, having this perception of something. And then there's people
Mhm.
who are like, the oscillations or the having this perception is because you have this like state and this like state of IE balance, and you know now the animal is more awake and it can like be better at perceiving blank sensory stimuli. Whereas if you go full labeled line on the other end, you're like, hey, these circuits are engaged, and because these neurons are doing all these transactions right now, you end up with a side product of like electrical activity that looks like these oscillations. You know what I mean? You can in both
Mhm. Yeah.
directions
Yeah.
end up with the same result, but actually you're recording. Do you know what I mean? Like it's because you're recording it different, and that's why we need diversity in kinds of scientists and the way
Yeah.
people think, because you can be recording the same brain area and like come to a different result just because of the tools you picked and
And
how
I
you
feel like,
framed the question. Yeah.
yeah. And I feel like um the like stimulation and intervention methods I feel like are helpful too for getting at some of these things too. Like with behavioral like inhibition versus excitation, I think like one of the interesting things is like if you just zap one half of the frontal cortex with TMS, you get changes in that. And like to me that like I find that pretty convincing that it's like the overall state of excitation is really what matters then, because it's like you didn't have to put dopamine on it.
Yeah.
I guess
Yeah.
people could argue like maybe it's selectively activating the dopamine terminals or something, but I think the general thought is that it's because it's making the neurons fire more.
Yeah, I was gonna say the only thing we have going for us in the labeled, I guess like the labeled line side of the team, it's all the I guess like old school like 50 like Penfield, where like you can
Mhm.
like electrically stimulate like small portions of the cortex that represents different parts of the body, and like the person is awake, so you can be like, what tingled, and they'll be like, that was my pinky finger on the left side,
Mhm.
and then you could like map a bunch of things on it. But even though, you know, like that's the closest we can get from like, you know, this place, Zach, this little place, and it, you know, labeled line represents like maps
Yeah,
topographically
and
to
it
the thing.
is like quite powerful. I mean, there's when they do surgery like on the hippocampus for people who have severe epilepsy, they'll do that, and people will be like, they'll zap an area, and then they'll be like, "What happened?" And people will be like, "I started thinking about my cat," and then like, like
There was like,
it's
don't go,
really like
that was a dark
like
place.
you can get a lot of weird, like you can get all sorts of things from just stimulating random parts of the cortex.
Yeah. And like we are very like I guess like the biggest, I don't want to say argument, but like example for me is like we have dermatomes, like we literally have stripes of
Mhm.
our body that represents
Mhm.
that like the neurons that innervate that patch of receptive field and that part of your body is like activating these populations of neurons. Those neurons go to like, you know what I mean? Like that is, it's very
Hmm.
patterned, like, and it makes some sense. It's not perfect, but you'll see cool things where animals that use a certain part of their body more, they'll have more representation in that part of the cortex. Like my favorite example is in the moles. The star nose moles with really like
Hmm.
the face thing.
Yeah.
They can
Yeah.
sense like a vibration from like feet away of like an earthworm shifting like a grain of sand, and so they can like feel the
And
direction
so
that
it's
there are
these
worms.
moles that have like fingers around their nose, right?
Yeah, yeah, they have they're called star nose moles. They have these like really super sensors on their face. And then if you like stimulate and record from their cortex, they have way more cortex that represents these little like fingers on their face.
Uh-huh.
Oh, you're putting a video
Yeah, no, I
of
just put the link so that people know what we're talking about. But
yeah,
they're
yeah.
they're pretty like like they're a little bit an SFW. They're like pretty freaky looking. But
Yeah, freaky
uh,
blind little critters, but like, famously they have these, super sensitive, star nose thing, and they have more cortical representation for that star, like more than their eye, like their face, the rest of their face, their bodies, like even their paws, like their nose gets like a big portion of the cortical space. And that makes sense to me, like that. Or I mean, like intuitively that means like, you need more neurons to represent this like, little spidey sense thing you got in your face. So you need more real estate um to like,
yeah.
handle the input coming in to that space. Um
Yeah, like in a lot of rodents, right? Like people notice that kind of their somatosensory cortex have these like weird giant kind of cylinders in it, and they were like, "What is this?" And it turns out it's
yeah.
like a specialized part of the cortex to handle the input from their whiskers.
Yeah, yeah. The barrel cortex is like famously
Uh-huh.
layered. Like the rest of the mouse brain, the cortex brain is like mostly like salt and pepper speckled. But they have barrel cortex that is actually properly like layered. And I say like actually proper, but like humans have all the layers most places, I think. So like basically the equivalent of the barrels in the mouse cortex is like what we have in all of our cortex, which is kind of cool. Like we have these columns, like the column organizations. We have like topographical organizations. It's quite nice. Like, yeah. So anyway, those are cool examples of, like in my mind, those are examples of labeled lines. But like, at the end of the day, maybe it's just examples of circuits. They're just
Hmm.
examples of like the groundwork. Like, like this is just how it's wired. Um,
Yeah. And I
uh,
guess like in terms of like a sort of a unifying idea, I mean, one of the things I do wonder about is like, okay, are labeled lines like I would kind of believe that the brain is mostly an ensemble coding system, but it uses labeled lines in some cases, and like a labeled line is just like I like kind of the way that I tend to think about it as like a labeled line is just an ensemble system that uses very sparse encoding. So like, and for like people who aren't familiar with that term, so it's kind of like any sort of complex neural network, when you're representing a stimulus, like it can be the representation is kind of spread out over like a million neurons that are, and each neuron just varies a tiny bit in how much it fires. Or it can be like you have one neuron that's just on or off depending on whether the thing is present or not. And like to me, it kind of makes sense to think about like, okay, maybe these really are not two completely different things, but it's just like the degree of sparsity of coding.
Sure. Like a really extreme example, like here are super reliable neurons. Like no matter what, you give it
Mhm.
a photon, it'll fire. And like these over here are, like there's like a range. And then there's these neurons here that will fire 60% of the time when it's given the light. And like that's the difference. Those
Right.
those unreliable dudes that are like like firing just barely above chance.
Mhm.
Like those seem like really hard neurons to study. And I
Yeah. Although they're really cool. So like there's some pretty good theories that like, for example, the way that we tell how far away, how far ago something happened, is that the receptive fields of neurons in the hippocampus just kind of gradually shift over time. And they shift at kind of a constant rate. So we can tell how long ago a memory was encoded approximately by looking at kind of how closely does its encoding match the encoding that we're currently using. Um, and to me, that kind of makes sense. Like I think in some, so having a very sparse encoding, I think in a theory, like in an information processing sense, is useful for some things. It lets you kind of essentially extract the different dimensions from stimuli. So for example, like you can have dimensions of like, oh, this is something that tends to be in my house. And then it helps you form associations. It's very useful for memory. But on the other hand, like you probably don't actually want distributed coding of like touch sensation that has to come from your foot, because then need like you'd have to have like 1,000 neurons all hooked up to the same patch of skin, and it makes more sense to just have one neuron that encodes like how hard is it being touched.
Yeah, I mean, even in the most strict, and I'm like really for trying to understand, how the
Mhm.
brain works, like from like the labeled line perspective. Even in the most strict version of that, like where it's like simple, like temperature, for example, it gets confusing. So like the ion channels that are needed to be activated for heat, for example, are expressed in many sensory neurons. It's not like tied to a specific small population. Like cooling, like menthol is in a small population that is unmyelinated, and they're like the size, and you can like histologically find them. But like temperature is difficult because, and even if you think about how we perceive temperature, like there is some crosstalk. So there's this illusion called like the temperature grill. So basically if you
Oh yeah,
have
the thing
like,
of the exploratorium.
yeah,
Yeah.
yeah. So like if you have like a cool like a pencil, and it's like they run warm water through it, and then you'll touch it and be like, oh, that's warm. And you have two pencils next to each other, and you can run cold water through it and be like, oh, that's cold. But if you like take like alternating like cold pencil, warm pencil, cold pencil, warm pencil, and you put them all together into like a grid, and then you touch it, you can perceive like hot, like heat, like pain. And it's just alternating hot, like warm and cool. Like either individually it wouldn't be painful, but like if you give it together, that illusion, you immediately get like a with you would pull your hand away and you would perceive like pain, and hot actually. And so like there is some crosstalk either at the spinal cord or higher up, where like this like purely labeled line thing doesn't it's not enough to tell the whole story, right? There is some crosstalk happening as it gets even on the ascending path, like as it goes into the spinal cord and into the brainstem, like there is some things modulating each other, and that is interesting, like
Mhm.
understanding how it gets modulated, what modulates it, like that's what keeps me up, and that's what I like want to study. um and that's from like a really simple thing where you're like, oh yeah, you understand how this works, you understand how these works.
Right.
Like,
Yeah.
wait, how did you what wires got crossed that you
Like
perceive pain?
it should be like one of the simplest like things that we know of is like
Yeah,
you
like we should
put
be like,
heat
ah, that is
on the
warm,
skin.
that is
Yeah.
warm and cool, but like somehow it just gets it goes straight into love. What the fuck, withdrawal? Like pain. Like it
Do we
does the
know
thing.
like is there a good theory about what is going on with that now? Or are we still just like, huh, that's weird.
I think we're I mean, there probably are people working on it. I haven't read deeply into it
Mhm.
recently, but yeah, I mean, like the solution was not discovered that long ago. Like I think Catherine Bushnell described it like in, I want to say like the early 90s or
Mhm.
maybe late 80s. I'm like trying to like figure out how old she was when she like came up with this assay. But yeah, and they like did this on human patients and put them in fMRIs, and they would like literally perceive it the same as when they like put their hand in like hot water. So like they're perceiving it really similarly. And yet the labeled line, like the actual neurons being activated, do you know what I mean? They don't do the thing. On the other
Although
hand, when
like
you electrically record from these neurons, like you can do one, like you can, it's really difficult to do in an awake patient, but you could technically like do skin nerve recordings. Like you could put an electrode into their arm and then like, give them a sensation, and then you can like record from individual neurons at a time. And these like through those kinds of experiments, you can kind of get like a real like a readout. They're very difficult to do.
And I guess like
go ahead.
And I guess like to me it seems like a big like kind of one of the things that makes this debate kind of tricky is like you have to specify like what levels and like what part in the hierarchy you're talking about too, because it's like even with that, like okay, you can say it's not a labeled line at the level of the skin or at the level of like the sensory ganglion, but then it could become a labeled line if, for example, like the result of that is just that it activates like a pain system in the somatosensory cortex, for example.
Like it could convert into a labeled, like everything that feeds into this like locus, and suddenly it becomes like, go do the pain, withdrawal, like we hate this phenotype. Yeah, it could.
Yeah, I mean that kind of happens like with vision too. For example, like our vision, we have neurons that get activated by like a dark surround and then a neutral center, or a bright center and a neutral surround, and they just kind of converge on the same system. And that system is actually kind of the thing that would be like the labeled line, even though it's activated by multiple different
That's
things.
true. And that's how we get all our edges and stuff. Yeah.
Yeah. But then on the other hand, like the part that I find more troublesome is like you're like, okay, and then it activates the pain system, and then it like releases pain juice into the brain, and like that's the part that like I don't believe. I don't think that it makes any sense to talk about like, oh yeah, this is like the neurotransmitter for pain. Like I think you have to conceptualize it as like this like the important part is the interaction between these different systems and like what it makes the animal do.
Yeah, studying pain juices is hard. Yeah,
like it goes to the pain system, which is where all the pain juice is.
Yeah, it's not even super well defined, like what is a pain area and what isn't a pain area. I find this really fast. So one of my favorite ways of thinking about this is actually to think about what happens when we definitely don't have pain, kind of like when you talked about consciousness, like we talked about anesthesia. I feel like a good way to think about pain is also like to like break down like why, and like we should just reverse engineer anesthesia. And
so like what is not happening when we don't have pain?
Yeah, because I mean the difficulty in that is that most anesthesias are just like broad GABA inhibitors. So like if you inhibit GABA, sorry. Yeah, increasing the amount of GABA inhibition. So then it's like not very specific because we have those receptors everywhere. Like yeah, it's interesting when you can like really like narrow down on a particular circuit or a popular population of neurons, which is that could be a more interesting way to look at.
Or like similarly to like with the consciousness thing, I think it's interesting if you can look at like where do different things converge. So for example, like what gets suppressed by both like acetaminophen and codeine or something like that. And then
oh yeah, that's great. That's such a good
yeah.
Oh, so the problem is that also like acetaminophen doesn't do anything to you perceptually if you're not already in pain.
Huh.
Right? Like not perceptually. I guess
like it doesn't make you feel like numb if you're not in
pain.
Pain.
Like you have to be in pain first and then it'll alleviate the pain. But like if you like let's say like you sprained your ankle and you take Tylenol, you feel better. Like if your ankle feels less painful. But if you had not sprained anything and you just randomly took Tylenol, like I don't think it's gonna help you feel that much better.
It will,
like not pain wise.
It will act as a mood stabilizer, but is that true of like opioids too? Like if you like, I feel like if you like give someone codeine, like it's not kind of cutting off like any of their other sensory input, right? Like it's just suppressing their perception of pain.
Yeah, no, I think you get euphoria from it as well. And
True.
I
Yeah.
think
Yeah.
that's the problem. The euphoria is like recreationally fun. The problem
Right.
with it is that I think because of how long drugs stay in your system, it's way longer than we naturally like our endogenous versions of like opioids like don't last that long. And so like because it's like these drugs are you when you consume these drugs, like a lot of times these receptors will downregulate so that
It desensitizes it.
yeah, so
Yeah.
it desensitizes it so you don't like go into like seizures and stuff. So your brain will have like this like a dynamic plasticity that would like reduce the amount of receptors there so that you know you don't go into as high of a high in the future. And so actually that's why you have to like usually when you take these kinds of drugs you end up needing to take more to get the same effect.
Mhm.
um and then you end up craving it a lot because now your brain is like we don't have this molecule. It's really
Yeah.
low because we expected it to be super high and we had down regulated these receptors. And that's like a common like issue with many addictive drugs, right?
Mhm.
It's like
So I
you
was curious what you were saying about so like the problem with using these drugs to study stuff is like kind of they don't necessarily have any perceptual effects unless you also have pain.
yeah, yeah, like
Yeah.
especially like the the over the counter ones, the aspirins, the aspirins and the acetaminophen and the NSAIDs. Like a lot of
Mhm.
those, like I wonder if it's like their primary action is not even on. Sometimes I'm like they're probably not even on the neurons. Like
Yeah, I mean,
they're
NSAIDs, I think we're pretty confident that the mechanism of that is mostly that it's kind of immune mediated. It's just reducing a lot of the inflammatory signaling at like an injury site, which is why they're actually better for like if you have swelling, it's better to take aspirin than it is to take acetaminophen because acetaminophen will make it not hurt versus aspirin will actually like treat the inflammation. Um, but acetaminophen is a super weird one, actually. I don't know if you've read about it, but it's like we don't really have a clear sense of what it's acting on. It seems like it's sort of a cannabinoid, maybe. Um, but it also seems to like dramatically impact emotional regulation.
that's so crazy. Like we take like you can go to Walgreens right now and buy a bottle of Tylenol and no one will blink an
Uh-huh.
eye. And we don't know exactly how it changes our brain and why we perceive painful things as less painful when you're on it. What?
Yeah. It's really great though. Like if you're really sad and like you take Tylenol, you'll be less sad.
Please do not overdose on Tylenol because it could cause liver problems if
Yeah,
you
but if you take like a normal high dose, like there's actual studies where they like gave people that like a normal high dose of it and they found it like made them less lonely. And
like it works. It's doing a thing. Like we should know what they're
yep.
doing, right?
Mhm.
Um, yeah, fascinating.
Mhm.
Anyway, this was really fun.
Yeah.
I
Yeah, this
knew I
was
didn't have to
fun.
prepare more for this.
Uh-huh. Yeah. Yeah. We
Thank you
can
for
just
joining us.
like yeah, yeah.
We're philosophizing. Come back for
Oh
dreams.
yeah, so next week we're gonna be talking, so Prachi is gonna be coming back, right?
Yes, Prachi's coming back, and we're doing
Yeah.
dreams next week.
Yeah, we're gonna be talking about dreams. So yeah, bring us your questions about dreams.
The dreams.
Yep. Great. Bye.
Bye. Have a good Friday.